Hi, everyone. Welcome to the behavior session for our Evolution 2023 virtual meeting. We have 4 speakers, 15 minutes each. When we get towards the end of the talk, if there's some time, if you click the little raise hand button at the bottom there, you can chime in with a question. You'll be muted, but we can unmute you. Okay, so our first speaker today is Adam Bode.
You hear me?
Now I can, yes.
Okay, great. And can you all see the slides?
Looks good.
Great. Everyone, my name's Adam Bode. I'm a PhD student in anthropology at the Australian National University. And the title of my talk today is Romantic Love Evolved by Co-opting Mother-Infant Bonding. Sorry, something's just happened. There we go. So what is this presentation about? Essentially, I'm going to introduce a top-down evolutionary model of romantic love, informed by the theory of co-opting mother-infant bonding, which tells us about the mechanisms involved in romantic love. And I'll get to these in detail, but essentially I'm going to talk about 3 systems that were co-opted from mother-infant bonding: the mother-infant attraction system, mother-infant attachment system, and obsessive thinking, and 2 systems that were combined with those co-opted systems: courtship, attraction, and sexual desire. Oh, is this working? I've got a pause.
Should be.
I've got something that's popped up here that, sorry, I'll just have to share again.
Okay.
Sorry, can you see that now? Why is this important? So there's 2 reasons. The first is that this theoretical framework can guide the mechanistic and psychological research in terms of future studies, but it also makes sense.
I don't think we can see your slides still. Now we can.
What a disaster. Okay, sorry, it'll go smoother from now on. So why is this important? Essentially, there's 2 reasons. This theoretical framework can guide mechanistic and psychological research, including ideas for future studies, but also making sense of existing findings. And understanding the evolution of romantic love tells us about how pair bonding evolved. The precursor to romantic love probably evolved first in conjunction with pair bonds, and pair bonds are defining characteristics of humans. So we understand how romantic love evolved. We understand how pair bonds evolved. So what is mother-infant bonding? There isn't actually a great biological definition, but there's a good psychological definition that's relevant. And Bicking, Kinsey, and Hupsey essentially said that it's a maternal-driven process that occurs primarily throughout the first year of a baby's life. It's an effective state of the mother, and maternal feelings and emotions towards the infant are the primary indicator. It's also very useful because it indicates that behavioral and biological indicators may both promote maternal-infant bonding or be an outcome of maternal-infant bonding. So what are the mechanisms of mother-infant bonding? There appears to be 3 general systems involved: a dopamine and oxytocin-rich attraction component characterized by activity of the left ventral tegmental area, right thalamus, left substantia nigra, and putamen; a dopamine, oxytocin, and opioid-rich attachment component, which is most characterized by circulating oxytocin; and an obsessive thinking component. So what is romantic love? Bode and Kushnick in 2021 defined it as a motivational state typically associated with a desire for long-term mating with a particular individual. It occurs across the lifespan and is associated with distinctive cognitive, emotional, behavioral, social, genetic, neural, and endocrine activity in both sexes. Throughout much of the life course, it serves mate choice, courtship, sex, and pair bonding functions, and it can best be described as a suite of adaptations and byproducts that arose sometime during the recent evolutionary history of humans. It's typically associated with the early stages of a romantic relationship, but can occur in the absence of a romantic relationship or for many years. So what is co-option? It's basically a process where a trait, such as a mechanism, morphology, or behavior, or most commonly in the literature, genes, are replicated in an innovative form to serve a different function to that which it originally served. So the commonly cited example is that of the feather of birds, which were originally believed to serve a role in thermoregulation, but have been replicated in an innovative form to play a role in flight. So there is one existing theory of an evolutionary theory of romantic love called Fisher's theory of independent emotion systems. It was postulated in 1998 before a single study of biological study of romantic love had ever taken place. It talks about romantic love as focusing energies on a preferred mating partner, and that system is associated with catecholamines, phenylethylamine, and serotonin. It's not a great model. It doesn't allow us to test it very well, and it doesn't explain a lot. So this is why this new theory is relevant. So the new theory of co-opting mother-infant bonding essentially says that some of the cognitions, emotions, behaviors, neurobiology, neuroendocrinology, and genetics of mother-infant bonding were co-opted to form romantic love. The function changed from mother-infant bonding to pair bonding, specifically pair bond formation, but other mechanisms in psychology, specifically courtship, attraction, and sexual desire, may have also been combined. So there's preliminary evidence in support of this. The first is that there's a lot of psychological similarities between mother-infant bonding and romantic love, such as altered mental states, preoccupation, longing for reciprocity, exclusivity of focus, idealization of the other, heightened sense of responsibility, proximity seeking, time-limited duration, and baby talk. So now I'm going to talk about the three co-opted mechanisms, the attraction, attachment, and obsessive thinking systems. So there's evidence that an attraction system was co-opted from mother-infant bonding to romantic love, and they come from two meta-analyses of fMRI studies. The first, by Ortiz and colleagues, found overlap in regions rich in dopamine and oxytocin in both maternal love and romantic love. And this is specifically the case in subcortical reward-related brain systems. A more recent meta-analysis found that the only region common to both maternal love and romantic love was the left ventral tegmental area, and both of these, and that is highly rich in dopamine receptors. So evidence that an attachment system was co-opted comes from a seminal study by Bartels and Zecchi, fMRI study. They found that activity in romantic love overlaps precisely with maternal love, particularly in regions rich in oxytocin and vasopressin receptors, such as the striatum, middle insula, and dorsal part of the anterior cingulate cortex. There's also high levels of circulating oxytocin in the early stages of a romantic relationship following interaction, but also in new parents following gazing into their infant's eyes in pregnancy and following mother-child contact. There's also high circulating beta endorphin in early stage couples and new parents following interactions than compared to singles. So there's evidence that the obsessive thinking component was co-opted as well, although we don't know anything about the biology of this process. But psychologically, having one's daily routine interrupted by thoughts about their loved one or infant is common to both. Thoughts about their infant or loved one interfere with concentration on other things is common. Visually imagining their infant or loved one when not in direct contact is common. And thinking about their infant and loved one when at work or otherwise engaged is common to both types of love. So now I'm going to talk to you about the two systems that I think were combined, the courtship attraction system and the sexual desire system. Romantic love serves a mate choice and courtship function. And we know that romantic love is associated with bilateral activation in romantic love, specifically VTA bilaterally. So this suggests that there's some other aspect of dopamine-rich activity associated with romantic love. And romantic love also serves a sex function, and sexual desire was probably combined with other aspects of romantic love. And there's similarities between sexual desire and romantic love, specifically activity of the caudate, insular, putamen, and anterior cingulate cortex. And there's also one study, although inconsistent with other studies, that found increased testosterone in females in love. And increased testosterone is associated with increased proceptivity, which is the desire of females primarily to initiate sex. So in sum, the model that I'm proposing is that there were three systems that were co-opted: attraction, attachment, and obsessive thinking, and two systems that were combined, courtship, attraction, and sexual desire, to form what we now call romantic love. So in conclusion, the model that I've proposed is internally consistent. It's supported by evolutionary theory, draws on the past 20 years of romantic love research, and is a vast improvement on Fisher's model, which can be tested and can guide research. It also demonstrates that a top-down evolutionary approach can inform the study of the mechanisms of romantic love. Thank you.
Thank you, Adam. Any questions from anyone? You can hit the raise hand or you can type them in the chat. We have a couple minutes.
Sorry about all the technical difficulties. That was all my fault. I started pushing buttons I shouldn't have.
No, you're fine. It worked out all right. Can I ask you, on the last slide, you mentioned the improvement over the previous model.
Yes.
And then you mentioned it was this new one is testable. Like, what would be like a really good, like, what would be the next thing you would do to sort of test that model?
So the two things I'd like to focus on is looking at the psychological similarities and differences between mother-infant bonding and romantic love. So I can really establish the cognitions, emotions, and behaviors that are common to the two. We should then be able to correlate those with specific neural activity, which would really open up what we know. I also think that PET scanning might be particularly useful because there's been to date 45 or so biological studies of romantic love, 30 of which are fMRIs, and about 25 of them all basically do the same thing. So it'd be really good if we could get some acute neuroimaging information, isolating those 5 individual systems that I said were involved, and demonstrating their individuality relative to the other 4 systems.
Nice. Any other questions?
Thank you very much.
Thank you. All right, so that we can stay, that's a hand clapping. I thought that was a raised hand. So we can stay on schedule. Let's give about 4 minutes before the next talk. So next up will be Brenna, and so you're welcome to share your slides when you want. But yeah, let's try to get going at 5:45, so we're on schedule.
Okay, I'll just wait a little bit. All right, should I wait another minute?
Yeah, let's give it 30 more seconds in case someone's migrating over from the other session. Okay, next up is Brenna Decker. Go ahead.
Hi, hello everyone. My name is Brenna Decker. I am a PhD candidate at Utah State University, and today I'll talk a little bit about one of my dissertation projects, the plasticity of Pompilidae behavioral evolution. So first I want to introduce wasp behaviors generally before I dive into specifically spider wasp behaviors. There we go. So wasps are in the insect order Hymenoptera, which includes 3 main groups: the Symphyta or the sawflies, a paraphyletic group called Parasitica, and then the stinging wasps, Aculeata. So the Aculeata wasps do not include the ants and the bees. And within these 3 main groups of wasps, we have a lot of different behaviors, from being vegetarians, carnivores and predators, and even scavengers. And then there's another group that we call parasitoids. There's ectoparasitoids and endoparasitoids, where ecto meaning the larvae actually develop external to the host, and endo where the host or the larvae develop internal of that host. We can go even more specific with this with koinobiont and idiobiont, where the first term states that the host continues normal activity after it's been parasitized, and idiobiont is when that host is actually permanently paralyzed. And then on top of that, there's also kleptoparasitism, where one wasp will actually use the host that was collected by another wasp to lay her own eggs and basically hijack that host. So Pompilidae are the spider wasps in the group Aculeatum. So they're named spider wasps because they exclusively use spiders as hosts, and all larvae develop on the outside of a single spider's abdomen, so they're ectoparasitoids. Many of the behaviors that these wasps use to actually get that host is to be hunting before or after excavating their own burrow or finding a pre-existing cavity, or in cases of the coinobiont parasitism, they don't use nests at all where that host resumes normal activity. So with all of those different behaviors, back in 1953, Dr. Howard Evans actually proposed a Pompilidae ethocline. So an ethocline is just an evolutionary hypothesis for the evolution of different behavioral patterns. So this ethocline that he proposed used some of the just basic behaviors that he at the time and others at the time that were researching pompilid behavior were actually using, and the different orders of those different behaviors. So within this chart, we have hunting as V, stinging or paralyzing the prey as P, transporting or carrying that prey to another location, T. I is for preparing some sort of nest cell or burrow. O is for the oviposition or the act of laying an egg on the host. And then some of these behaviors end with closing that nest cell or burrow. So as an example, one of the predicted ancestors of pompilids had a behavioral pattern of hunting, paralyzing, and transporting a host, and then after transporting, they laid the egg on the host. Within pompilids, one of the behaviors that we see is actually constructing a burrow first before hunting, paralyzing, and transporting that host back to that burrow. And then once it's at that burrow, ovipositing or laying the egg on the host, and then subsequently closing that burrow behind it. When he did this ethocline, Evans lined everything up specifically from simple to complex, with 2 reversals to what he labeled as more simplistic behaviors. Within this same publication, Evans did explain several morphological characters that related to some of these behavioral patterns that were being observed at the time. So I want to go through whether, based on that ethocline 70 years ago to today, when we have some more information, I want to see how well that linear progression from simple to complex fits our actual behavioral patterns for pompilids. So the Pitts lab has been extensively sequencing ultra-conserved elements, which are just 160 to 180 base pair segments within the DNA that are very conserved throughout the animal and plant kingdom. And we constructed a new molecular phylogeny for pompilids. So I'm using a test subset data to see if whether we have the bathyloid scalioid sister group for this updated phylogeny, and if we can see any of the behavioral changes that are present in that ethocline. So specifically, I was looking for a pattern from closed cell kleptoparasitism to open cell kleptoparasitism, from trapdoor hunting spider hunters to temporary paralysis, and then seeing if the behavior of mud nesting is the end behavior of pompilids. Okay, so the first thing that we can address is that we know there is a different sister group to pompilids. So instead of the bathyloid scalioids, we have a Mutillid sapigid sister group to pompilids based on more recent molecular work. And the interesting thing about the Mutillid sapigid is that we have open cell kleptoparasitism. And we also have a lot more information, but we had to still fill in the gaps for some of these behaviors of these pompilids. So first, these are all anecdotally related. There's no publication on morphology and linking to behaviors for pompilids. This is just what my advisor and I have observed through our studies. So hind tibial teeth and foretarsal combs are usually characters that allow for digging. So either digging already closed cell or digging in new burrow before or after hunting for the spider host. Non-diggers do not have those characters. Then there's also the mud nesters or cavity modifiers, where one genus actually has a flattened spot on the abdomen, but then other groups of pompilids actually have the setae on the underside of their mandibles. And then there's trapdoor spider hunters or enterers, I was calling them. They have these enlarged forefemora, so they can actually physically pry open those trapdoor spider burrows. And then they usually have the very flattened pronotum and head shape, so they can easily slide into the burrow and paralyze those spiders. This morphology, the head and the pronotum, can also be seen with any other spider wasp that enters some sort of spider dwelling, not necessarily trapdoor spiders. Okay, so what we did was I made a character state matrix of all the species that were included in this test data and use that morphology to give them a letter. So the letter for the behavioral pattern is going to be different from Evan's ethocline, just because there are actually a lot more behaviors that we have discovered in the 70 years since he proposed that. Because some of the wasps do not have a lot of defining morphological characters, I gave those up to 4 different possible behaviors if we did not have any publication relating to that specific species behavior. Okay, so with our sampling, we got 347 taxa that were included. 21 of those species were based on publications, but the rest of the behaviors in this test study were estimated based just on the morphological characters from that character state matrix. So we use base traits, multi-state, MCMC analysis with reverse jumps to get an average, at least 1,000 iterations, and then we average those rate changes from one behavior to the next. So this is a very preliminary ethocline based on that analysis. First, we have one of our outgroups, which are grub hunters. They change to this second outgroup and basal pompilid group with the open cell kleptoparasitism that goes to closed cell parasitism, and then trapdoor enticers. So they actually strum on the silk, entice the spider out, and then paralyze it, and then actually bring it back into its own burrow. But from here, it gets pretty crazy with just arrows going kind of everywhere, and then out of nowhere, we get cavity modifiers and mud nesters. Nothing is actually leading into that system, so I'll explain that in a little bit. But I do feel like these rate changes are really conflating the actual rate changes that are actually in existence. So I want to go through those. These green arrows are rate changes that were above 0.2. So the rate changes that base trait outputs is between 0 and 1. So all these green arrows are just 0.2. Then we have the blue arrows that are rate changes between the 0.1 and 0.2, and then I included these red ones that were below 0.1 because those were the only changes from the behavior to another behavior. So the typhoids, the grub hunters weren't even really included in the final output. So because we had them in the phylogeny, I put them in and they only had a 0.02 rate change to open cell kleptoparasitism. So I did that with all the other behaviors that didn't have very high rate changes. Okay, so looking back at Evan's ethocline, I am very hesitant to say that we have a final ethocline, but we can look and see that there are some generalizations that we can make. First, we can reject most of our hypotheses. The first, obviously, we have a Mutillus epigis sister group and not the Bathyloid scalioid sister group. And most likely the ancestral state is open cell kleptoparasitism. Then one of the trapdoor spider enterers does actually change to a temporary paralysis with that spider wasp actually entering the spider's hideout. So that's close, but there's also coinobiont paralysis where the spider actually gets up and moves and it's just open air paralyzing. And then mud nesting, there's a back and forth between the cavity modifiers, but it's kind of weird with that. So we need to do a lot more analyses. So again, there's a lot of behavioral options with those that didn't show a lot of morphology. So we are adding about 800 plus more samples, collaborating with global professionals and estimating the behavioral states at each of the nodes. So with that, I'd like to thank you all, especially my committee members and the department for helping me with this project. Thank you.
Thanks, Brenna. We have time for one question. Anyone have a question? So I'll ask you something because I'm curious. It seems to me like it would be really, really hard. And what's the approach for like coding all the characters, right? Because they're so complex and nuanced, and like deciding when you call it this versus that seems like it could get into a real challenging.
It can, and I even have subcategories for each of those as well. And then what I did was, if it was just present and absent, we're going to mark it this way. If there's nothing, we are giving them all 4 options for the behavioral pattern. And I was doing that a lot because a lot of the females do not have those morphological characters. So that's why it's going to be really important to do a lot of collaborating and see if there's just observational data out there and see what we can compile and see if we can narrow that down a bit more.
Right. Super cool. Okay, so let's it's 6, so let's move to the next talk. So next up we have Simona Kelly.
Yes, let me share the screen. Just a moment. Okay, so hello everyone. I'm Simona Kelly. I'm lecturer at St. John's University in Rome, and together with Martin Brune, who is a professor at the Ruhr University in Bochum, Germany, I work on this review on laboratory studies on schizotypy that will appear later as a chapter of an edited book on this very topic that is schizotypy. As I was saying, we run this review starting from the famous 4 Tycho Timberger's questions, and we try to value the complexity and heterogeneity of threats related to schizotypy. The study of schizotypy can be traced back to at least 1 century ago, when authors such as Bleuler noticed that there were recurring threats referring to oddity and centricity among healthy relatives of those diagnosed with schizophrenia. Moreover, they noticed how these very threats were also associated with premorbid states, leading to psychosis and schizophrenia itself. So the schizotypy refer to a very wide Latin personality organization that is consistent with an idea of continuity of traits and experience that may range from subclinical or even healthy states, even referring to creativity, so to an effective trait, to extreme states of mind that might be related to personality disorders such as schizotypal or schizoid personality disorder, and then psychosis and schizophrenia. For a more descriptive point of view, modern literature suggests how schizotypy would comprise at least 3 features. That is, positive feature referring to, for example, marginal thinking, negative, such as anhedonia, and disorganized features such as odd behavior or thoughts. This idea that, as I said, emerged about 1 century ago is highly consistent with modern approaches to psychopathology from a dimensional point of view. I'm talking about models such as 5-factor model that's, for example, tested also in non-human animals, and alternative model of personality disorder. This idea can be also considered consistent with an evolutionary look and human personality, and refer to an extremely prevalent trait, since recent studies suggest how 1 out of 10 persons would present these traits, again, ranging from healthy to maladaptive states. When summarizing evidence, we focus obviously on diverse evolutionary premises, but at least the 3 you can see here. It is the idea that sexual selection is pivotal in understanding human evolution, and then when dealing with the complexity of phenotypic evolution, understanding the correlated evolution among traits is important. And finally, we explore the role of life history theory that has been extensively applied to the study of human personality from an evolutionary point of view, even if its reliability is quite disputed. So, as I said at the beginning, we had this initial framework that is the one of the Fortenberger's question, that is the intersection of 2 dichotomous questions, proximate and ultimate explanations, together with either a diachronic or synchronic perspective to the adaptiveness of the threats. So the kickstart of any kind of evolutionary consideration about schizophrenia is that schizophrenia and schizotypy, since the 2 construct neurophysiologically are quite overlapping, are really a puzzling paradox. Since on the one hand, we know that schizophrenia has been presented in all the eras of human histories and all the culture, but at the same time, they have a huge evolutionary cost. So in this sense, both schizophrenia and this underlying personality organization defined as schizotypy is really a paradox from an evolutionary point of view. The evidence collected are quite diverse, and as I said, mostly they have been collected talking about schizophrenia, but since the 2 constructs are quite overlapping, we can integrate information from the one and the other. So from ontogenetic and genetic point of view, we know that there are several developmental stability models positing that schizophrenia may result from a failure in buffering the negative effects of diverse pathogen, toxin, and so on. And also we know that there is robust evidence suggesting how schizophrenia is highly hereditable, even if we have to admit how no single locus has been associated with a less moderate effect size. At the same time, we know that several social aspects are pivotal in understanding the emergence of schizophrenia and schizotypal traits. For example, social isolation, migration, change in social status, or breakup of romantic relation are associated with an increased risk for schizophrenia. On the other hand, immunological aspects has been studied suggesting how aberrant neurodevelopment secondary to viral transmission may be related to the later development of schizophrenia spectrum disorder, that is of a clinical manifestation schizotypy. Quite interesting, that is, for example, the study about Toxoplasma gondii and the study on rats showing suicidal behavior or definable as suicidal behavior, at-risk behavior. And when studying the presence of the Toxoplasma gondii, for example, in humans, we know that those reporting Toxoplasma show a higher risk for manifesting psychosis than general population. From a phylogenetic point of view, we have a wide and diversified elements that may help us understanding this paradox. First, we know that, for example, there are symptoms referable to human psychopathology, such as depression and anxiety, are studied and presented in diverse non-human animals, but this is seemingly not true for psychosis. At the same time, there are recent interesting studies showing, for example, that anatomically model humans that have been diagnosed with schizophrenia show less Neanderthal-derived genetic variation than general population. And moreover, having more Neanderthal admixture seemingly predicts less severe symptoms when diagnosed with schizophrenia. All the studies seem to suggest that schizophrenia emerged together with the rise of modern humans and possibly modern human social brain. Indeed, we have neuroscientific evidence, for example, about evolution of neurocortex in human lineage, suggesting how there have been a sort of evolutionary trade-off across different regions, such as amygdala, between greater information efficiency and reduced robustness. This latter exposing modern humans to an increased risk of psychopathology when compared to, for example, apes. At the same time, we know that the schizotypal traits are related to a genomic region, especially the prefrontal cortex, that are associated with higher human functions such as mentalizing. And so they may have exposed phylogenetically humans to a strong selective pressure and finally to a higher risk for severe mental disorders such as the one related to the schizotypal continuum. Making a step forward and going to the evolutionary model of schizotype itself, the first one is possibly the one by Price and Stevens. They suggest that in 1988, how schizotypal trait would have played a role in promoting sort of divergent thinking, and this can be associated with a higher proneness to religious leadership, generating new ideas, and also to a behavior related to the dispersal of human community due to the tendency of group splitting. More recently, the judiciary have elaborated some early intuition by Nettle, suggesting how sexual selection will have played an important role in the emergence of schizotypal trait, and this can be possibly linked to a short-term promiscuous and possibly polygamous mating strategy consistent with the life history theory model, where we do have a fast and slow strategy. This evidence at the same time have been reputed inconsistent for authors such as Shainer, that they consider instead symptoms related to schizophrenia and schizotypy as a low fitness variance of sexually selected traits. In a recent paper I have published early this year, I have tried to put together what we have said, especially about the continuity between healthy traits such as openness to experience and intervention and more maladaptive manifestation. So the idea is that this threat will represent a sort of vulnerability to adverse breeding conditions, including childhood maltreatment. And so these personality traits would be associated or have evolved in schizotypal trait. This can reduce the capacity of a person to somehow socialize a creative potential, its creativity. And so we may consider two maladaptive strategies: a risk minimizing strategy related to what is called as emotional and social detachment, that we know has been associated with a high level of intervention, and a uniqueness maximizing strategy, what is called, from a psychopathological point of view, a psychoticism, that again is associated with a high level of openness to experience. Psychoticism refer to odd behavior and thoughts. So the idea is that we may confer both the presence of a possible advantage related to creativity, but also its disappearance. And so due to these maladaptive patterns leading to a state where this creativity, this potential become disadvantageous. It is not the focus of this presentation, but just to let you know, we have used this evolutionary model to inform a psychotherapy for those reporting schizotypal tests, and this model have been tested successfully to a first pilot randomized control trial, in a few cases series, and now a new confirmatory randomized control trial is undergoing. Concluding, what we know is that schizotypy and schizophrenia is for sure an evolutionary paradox, is quite ubiquitous in time and culture, but it has huge evolutionary costs. Several hypotheses, several models have been proposed, even sometimes quite contrasting, leading us to the suggestion to promote and foster new research in terms of valuing the complexity and heterogeneity of schizotypal traits, and the idea that they can be better understood by considering a continuum between healthy states, such as creativity, and extremely maladaptive states as the one related to schizophrenia. So I have concluded. Thank you.
Thanks, Simone. I think in the interest of time, we'll have to skip questions. If you have a question for Simone, that you can put it in the chat or send him a direct message through the chat. Okay, so our final speaker today is Nicole Crianza.
Hi, everybody.
Hi.
All right, did that load full screen?
Looks good.
Great. So I'm going to talk today about the cooperative breeding and the evolution of birdsong. And this is work that I've done with my former grad student and now briefly postdoc, Kate Snyder, and Elena Loughran-Pierce, who was in our lab as a high school student, which was really amazing. And I'll be chatting with you. So the kind of driving question that we think about in my lab is, how is birdsong affected by selection pressures? So birdsong in the Austrian songbirds is a learned behavior. It's transmitted culturally. So birds need to have an adult tutor of the same species, usually in order to produce a song that's attractive to mates. So to think about the evolution of song, we have to think about its function and its role for birds. It usually functions in territory defense for territorial birds, species identification, and mate attraction. So in general, song is allowing birds to find potential mates, identify the right species, and assess the quality of those potential mates. So Darwin actually had this prediction that if birds with the best songs can attract the best mates, then sexual selection for showy and complex might favor showy and complex songs. And so within a population, there might be some metric that females are using to assess males that can sing the best and have the best song. So by extension, if birds are trying to attract multiple mates, such as in polygynous species, we might expect that the selection pressure would be amplified. And so as a result, we would maybe predict that polygynous birds should have more complex songs. And so this was something that had been studied in focal groups where they were both polygynous and monogamous species, but it hadn't really been studied across the phylogeny of songbirds. And so the syllable complexity in this sense is quantified in multiple ways, but the two ways that we quantified it were in syllable repertoire size, so the number of unique syllables in a bird's song. So here there would be 10 unique syllables in this song. And in song repertoire size, the number of unique songs in a bird's repertoire. So just in this little snippet, we would see 2 songs. And so to think about how song evolves kind of in parallel with the evolution of mating systems, and we compiled the largest database of mating systems and song features to date. And so we took published estimates of monogamy and polygyny, and later I'll talk about cooperative breeding, and we put those on a phylogeny, and then we also mapped on syllable repertoire size, song repertoire size, and a couple of metrics of song performance like intersong interval, song duration, and song rate. And so we got these data from a lot of different sources. We have the Birds of the World database hosted by Cornell Lab of Ornithology. The web of, we just mined the literature a lot. We put together existing databases, and we took papers that had already talked about and categorized polygynous and monogamous birds. So we wound up with 764 species where we could classify them as socially monogamous or socially polygynous. And we found that across all of the birds that we put together, songs were not notably more complex in polygynous species. So polygynous species did not have significantly larger syllable repertoires or song repertoires once we accounted for the phylogenetic relationships between the species. However, we did see that syllable repertoire size seemed to evolve faster in polygynous lineages. So this is an analysis called Brownie from Brian O'Meara's paper. This looks at the predicted rate of evolution of this trait if monogamous and polygynous birds evolved equally, if we didn't predict there to be any, if we didn't give the algorithm any difference between them. But if we calculate the rate of evolution where we allow monogamous and polygynous birds to differ, we find that the polygynous birds evolve, their syllable repertoires evolve faster. But since we don't see any systematically larger syllable repertoires in these birds, we predicted that they might just be kind of in a Red Queen's race where they may be oscillating in a pendulum, that they want to be different, but not necessarily always more complex. So we predict faster evolution, but not in a uniform direction. And we have these results published in Nature Communications. Polygyny is linked to accelerated birdsong evolution, but not to larger song repertoires. So then with the high school student that I mentioned, Elena, we were really interested in thinking about what about the opposite prediction? So what if sexual selection is not amplified, but instead kind of diminished or less male biased at least? So one scenario where this might occur is in cooperative breeding. So cooperative breeding is when there's the upbringing of offspring by adults other than the breeding pair. This is relatively common across birds, approximately 13% of songbirds and 9% of all birds. Still rare, but not so rare that we can't trace its evolutionary history. And people have predicted for a long time that cooperative breeding could be beneficial if because of kin selection, where if helpers are improving the survival of their relatives, and they're more likely to have surviving offspring if lots of individuals are helping than if every individual tries to make its own clutch, that there could be kin selection favoring this type of cooperation, particularly in places with harsh or variable environments. So the predictions here are kind of opposite to the ones that we saw in polygyny, where in cooperatively breeding species, we might predict that a proportion of males might opt out of the mating pool, decreasing the sexual selection on breeding males or increasing the selection on breeding females. And so by extension, we might think that song could evolve more slowly and females might be more likely to sing. So when we balance these selection pressures, instead of seeing accelerated rates of song evolution, we might see slower rates of song evolution. And we also might, in scenarios where the sexual selection is more equal on the two sexes, we might be more likely, our prediction was that we may be more likely to see females singing as opposed to male-only territorial song. So we again synthesize a lot of databases. We took the same data that we used for the Nature Communications paper and then added as many more species as we could find since we published that. And then this cooperative and non-cooperative breeding database we put together based on other published studies, and then by mining Birds of the World, Google Scholar, Web of Science for papers studying cooperative breeding in different species. We mapped this onto a phylogeny of all bird species in songbird clade from birdtree.org, that's Yates et al. And we merged our cooperative song breeding database with our song feature database that we used in the previous analysis. And then we took a really important female song database that was published originally by Karen Odom and colleagues and updated a couple of years later by Webb. We've updated that with, they put out kind of a call for people to study to document female song as much as they could, because it's definitely understudied. And so we tried to update their database with all the new information that's come out since then. So if we ask, do songs evolve differently in cooperatively breeding birds? We see once again that just if we look at the song features themselves, they weren't significantly different. Our sample sizes here are smaller, a little bit smaller than they were when we're just looking at the song features. So we don't have that many cooperatively breeding birds in this database, but systematically they don't have statistically different repertoire sizes. However, if we do our analysis again, where we look at the rate of evolution, we again have the dashed line as the predicted rate of song feature evolution if we don't know the difference between cooperative and non-cooperative birds. And so then if we map on cooperative breeding and non-cooperative breeding, we see the opposite result that we saw in the previous analysis, that cooperative breeding leads to slower song evolution, where polygyny led to faster song evolution. This is for song repertoire size, so the number of unique songs that a bird sings. So if we think about whether songs are evolving differently, we predict that their repertoire size and potentially their song complexity appears to evolve more slowly when birds are breeding cooperatively. Then we wanted to ask, does song repertoire show correlated evolution with cooperative breeding? So for correlated evolution, we use Bayes traits, like was previously talked about in this session. We have two traits that we can think about as evolving independently, and we can measure the rate of transition between non-cooperative and cooperative independently from lower song repertoire and higher song repertoire. But if we assume instead that one of these traits can influence the evolution of the other, we try to see, is there a significant relationship between the transition rates and the traits, implying that cooperative breeders might have different rates of evolution of their song, or that birds with different song repertoires might have different rates of cooperative breeding evolution. And so when we run this Bayes trace analysis and predict that there's correlated evolution, not independent evolution, we find significant prediction that this model is a better fit. So our kind of the first thing you'll notice is that cooperative breeding is lost much more quickly than it's gained. So the arrows, the thickness of the arrow indicates the rate of evolution of transition between these. So the cooperative breeding, regardless of song repertoire, transitions very quickly to non-cooperative. It's not a super stable state. But within the cooperative breeders, we see again the opposite result that we saw with the polygyny, that the in cooperative breeding, song repertoire tends to decrease. So cooperative breeding is lost, but within the cooperative breeders, we find that the song repertoire and the song complexity tends to decrease. So our answer to this question was that there does seem to be correlated evolution. We find significant predictive power of the correlated model, and that song repertoire appears to get smaller in cooperative breeders. And then finally, we had this prediction about female song. So we asked, are females more or less likely to sing in cooperatively breeding birds? So just looking at the raw data, there seemed to be a trend, so we were excited about it. Female song appears to co-occur with cooperative breeding before we accounted for phylogenetic relatedness. Cooperative breeders, we had 125 species that were cooperatively breeding, and 4/5 of them had female song, where in non-cooperative breeders it was kind of less than a half. So we got a lot of species where we could have both of these data. We're much more highly powered to detect differences rather than song features. So we had over 1,000 species with both data types. That was really exciting. And when we, but really what we wanted to do was think about the evolutionary history, not just the raw numbers. So here's a phylogenetic tree of these 1,042 species, where on the left, non-cooperative and cooperative are in red and blue, and on the right, female song absent and female song present are in red and blue. And our goal then was to say, is there correlated evolution? Are the red species more likely to be red or blue? Essentially, is non-cooperative breeding associated with female song absence or presence? And so when we do our base trace analysis here, we find that female song tends to be, is significantly predicted to be related to cooperative breeding. So our first trend again is that that cooperative breeding is very likely to be lost. It's not super stable in the birdsong tree. But that once a bird species is cooperative, it's more likely to gain female song. So we have a really thick arrow here in the cooperative breeders pointing from female song absent to female song present. So the transition toward female song is more likely to happen in cooperative breeders and non-cooperative breeders. And so we wanted to think about the same pattern, but across the tree, the whole tree. So is the transition to female song and cooperative breeding correlated in the lineages as well? So what we did here was did simulated character mapping, stochastic character mapping along both trees. So we made what's called sim maps in Phi tools made by Liam Ravel. And this was a method that we adapted from Wilson-Beck et al. 2003 to ask, do female song and cooperative breeding overlap in evolutionary history? And so this is a histogram of the amount of overlap, where we see that there's more overlap in the real data than in simulated data. So we did this thousands of times and tried to figure out if we simulate stochastic character maps with the real data versus randomized data, where we just randomize the tips, do we see more overlap in the female song and cooperative breeding than we might by chance? And so, and our results point to that we do, that in evolutionary history, female song tends to co-occur with cooperative breeding. So when we simulate these ancestral states, we find this co-occurrence. The p-value is tantalizing. P is 0.06. But we'll think about that a little harder. So when we ask, are females more likely to sing cooperatively breeding birds, we, in a couple different analyses, we point to female song appearing to evolve more readily in cooperatively breeding birds than non-cooperatively breeding birds. So I'd like to thank all the students that participated in this work and Join My Lab, our funding sources, and I'm happy to take any questions in the last minute or so. Thanks so much.
Thanks, Nicole. Any questions from the audience? Well, if no one else is going to jump up, I'm really curious about something, because
Cool.
is it the case in the Bayes analyses that the transition to a simpler song repertoire and the transition to females starting to sing, both those things are more prevalent whether they're cooperative or non-cooperative, right? It's just that the magnitude is bigger when they're cooperative.
That's right. That the transition seems to happen more readily.
So the repertoire seems to be shrinking generally, at least amongst the birds you're looking at.
Yeah, I can kind of pull that back up.
Which I know is unrelated to your main question.
No, it's interesting though. So the larger song repertoires, we have a separate paper that I didn't talk about the evolution of repertoire size and song complexity, thinking about kind of when that might evolve, and I think all of these things seem to a lot of these things seem to kind of tie back to female preference, that if the females are selecting birds with larger repertoires, the repertoires get larger in that lineage. And if they're selecting for more accurate learning or better song performance or territory quality or something else, then I think because the large song repertoires are hard to learn, hard to maintain, they get lost unless there's kind of a selective force, favoring them. So I do think that jives with our previous results, that you keep a larger song repertoire only if that's what's being chosen among the females in the population.
Cool. All right, well, thank you. And thanks to all the speakers.
Thanks everyone for listening.
Yeah, thanks to all the speakers in the behavior session.